Okay, so if a sperm and egg each had 46 chromosomes, and we learned yesterday that that's not true, hopefully, how many chromosomes would they have? That's the question, right? No, I'm sorry. How many chromosomes would a fertilized egg have? Sorry about that. See what you guys told me. Hopefully we got a big number, right? Okay. So it would have 92 because you'd be adding 46 and 46. Well, if you never were able to get back to half of your number of chromosomes in sperm and egg, eventually you just keep doubling the number of chromosomes you have, and that's impossible. That's exponential. That doesn't work. We know that. So we must, you should have your notes out. Your Chromebook should be on the table in front of you. You should be tucked into your little cubby. There you go. All right. Get cozy. I'm going to be here a while. Not a long time, but a small portion of your life. Oh, and now my thing's not working. All right. So it is 92. So today we're going to look at what happens during the first stages of meiosis and how the function of mitosis is different from meiosis. We might get into that today, but I'm going to focus mostly on meiosis today. And we might get into the mechanisms of genetic variation. We'll talk about one called crossing over as we talk about meiosis, but we will not quite get into all of them like independent assortment until tomorrow. So looking at meiosis, it sounds an awful lot like the word mitosis. And in fact, it is a form of cell division. But it produces daughter cells that have half the number of chromosomes than the parent instead of the same. You remember in mitosis, we started off with 46 chromosomes, and we ended up with 46 chromosomes. Well, in meiosis, we're going to end up with 23 in each of our cells. Now, during meiosis, a diploid cell goes through 2 divisions to form 4 haploid cells. Does anybody remember from yesterday the name of the diploid cell? What is the name of that diploid sex cell called? Oh, my word, my Bluetooth is not working today. Having some serious computer issues. Epic failure today. Huh. Oh, wow. No, we're not going home. Anybody remember the name of those diploid sex cells that go through meiosis? The ones that divide to make gametes. What do we call those? They're not the product of fertilization. That's a hint. What are those diploid 2N cells that we start with to make gametes? To make, what do we start with before we get sperm and egg? I'm slowly eliminating all the wrong answers. They're not somatic cells. They are germ cells, not somatic cells. Uh-oh, I'm going to sneeze. Baby. You ever get one? Yep. Oh, no, I'm not going to. Don't you hate that when you're like, oh, I'm going to sneeze, but then you don't, and so you're like your whole body gets ready for a sneeze and then nothing? Yeah, yeah. You got that release that never gets out. Terrible. All right. So yes, germ cells. I'll tell you, my computer is not liking me today. All right. This is where I need to draw these stages with you. All right, you guys. So yesterday we talked a little bit, and since my computer's acting so goofy, I'm going to walk right up to the board. So we start off, and you might want to draw this out with me, with a germ cell. And remember, you find germ cells inside of gonads. What are gonads? Ovaries and testicles. This is where you produce sperm and egg at. Now, although it's very different in boys and girls, the same concept is there, and the same division of the nucleus takes place. So a germ cell is 2N. Germ cell is 2N. Which means it has a complete set of homologous pairs. Okay? So it goes through two different divisions. The first division is known as meiosis 1. And this is where it goes from being 2N to 1N in two separate cells. What's 1N plus 1N? 2N, right? That's meiosis 1. Then, what? That's, for some reason, this thing is acting up quite disturbingly today. There we go. And then there's a second meiotic division called meiosis 2. Why do you do that? Meiosis 2. Okay? And this time, we're going to stay 1N. So let's talk about what happens to one homologous pair as we go through in each of our cells. I am going to make these, well, we'll do this. So we start off in one germ cell as being 2N, right? And I'm only going to follow one path, kind of, in a way, okay, with the chromosomes. So when they divide, in order to go from 1N to 2N, I would need to cut my chromosome number in half. So that means one chromosome will go here and one chromosome will go here. And that's at this point, where they're both 1N. Does everybody understand that? So we have a pair of chromosomes, we separate the chromosomes. Here's the question I'm going to ask you. Did I separate the chromatids? No. So the next division, what's going to happen is we're going to get separation of the chromatids. You guys understand that? So now I'm 1N here, 1N here. I was 2N when I started, and each of these at the end will be 1N. This is meiosis 1. This is meiosis 2. Which of these, meiosis 1 or meiosis 2, is the most like mitosis? What do you guys think? 1 or 2? 2. Here's why 2. 2, does my chromosome number change? No. I'm dividing my chromatids in 2, right? 1 is completely different. 1, I'm actually separating my chromosomes, cutting my genetic information in half. Big difference. Big difference. So meiosis begins with a diploid cell that has copied its chromosomes. Just like at the beginning of mitosis, except some very different things are going to happen. During prophase 1, the chromosomes condense and the nuclear envelope breaks down, and homologous chromosomes pair up. Chromatids exchange genetic material in a process that is called crossing over. Now in metaphase 1, The spindle moves the pairs of homologous chromosomes to the equator of the cell, and the homologous chromosomes remain together. Now, we are going to talk about each stage as we go. You might want to have an area or a piece of paper or something to draw these out. I'm going to have you draw them later. Oh my Lord, my phone keeps ringing today. Hello. Yep, Jacob Gilman, come on up. Your mom's on the phone. Yeah. It's Mrs. I want you guys to think he's getting yelled at by his mom. Oh, that would be scary, wouldn't it? Yeah. Well, I wouldn't do that. So prophase 1, right? They are going to be 2 divisions. So I'm going to have prophase 1, metaphase 1, anaphase 1, telophase 1. Then I'm going to have prophase 2, metaphase 2, anaphase 2, and telophase 2. So how many stages are we talking about here? 8, right? So mitosis was 4. Now we have 2 mitotic divisions. So we're going to go through each phase kind of together. Not kind of, that's what we're going to do. Prophase 1, what happens? Well, we've got a cell. There's a nucleus that's going to start to break down, and I'm going to start to see, let me give these chromosomes some different colors, okay? We're going to start to see our pairs of chromosomes buddy up. Did our chromosomes pair up in mitosis? Everybody say no. No. Okay. Let's do 3 pairs, just like we did with mitosis. We'll do one more. If my computer can handle it. Jeez. Oh my goodness. All right. How many chromosomes total are in this cell? 1, 2, 3, 4, 5, 6, right? Okay. All right. Now, I want to talk to you about one pair at a time because something special happens here. I'm going to look at this pair. I'm going to pull them over here to the side. And instead of making them both blue, I want to actually change one of their colors to green for a moment. Because when they line up side by side, something very unique happens. They actually swap genetic material. And this is what is called crossing over. So our DNA, because it's so similar, those 2 sister chromatids, they can actually flippity-flop with their neighboring chromatid. So these 2 blue ones were identical, right? But the second they trade information, are any of those chromatids alike anymore? No. So, men, you're on this planet for, let's say, 80 years. After puberty, well, even before, you'll be making sperm like 4 million a day for the rest of your life. You will never make the same one twice, ever, statistically. Because of this process called crossing over. Females, you'll never have made the same egg. You only make like 400 to 800,000, and you have them your whole life. But none of them are the same. This is why you will never have the same person unless you're from the same egg and the same sperm. It's called an identical twin. That's different. So every time they line up in this structure right here called a tetrad, they swap genetic information. Why do I call it tetrad? Tetris is a game of four-sided figures, right? Tetra, okay? Tetrad, 4 chromatids. So now, every one of those chromatids is different, and they will all eventually be in their own cell. How many chromosomes do we have total? 46, right? And we have 92 chromatids. Not any of them will be like the other one in the end. And then you get this whole new combination in every sperm and egg. Do you now start to understand how, with 30,000 different genes that we have, how we'll never have the same person twice? Think about the exponential on that. So, just thinking about the math behind that is mind-boggling. Okay, that's a matricy, right? It's like to space and back. So you will never have the same chromatid twice, folks. If I ask you where the source of variation in biodiversity is, that's where it comes from. Okay, so not only do sexually reproducing individuals mix their gene pool up by mating and things of that nature, but also within your own gonads, you have the ability to create even more diversity, different versions of you. That's why when you say you're half your mom and you're half your dad, the answer to that is kind of, right? You're half of parts of your mom and half of parts of your dad, because you're a different combination of who they are. That's a tough concept to grasp. Now, the other thing I didn't talk about is our centrioles show up, right, and form our centrosomes. This is no new thing. And they start to form spindle fibers, right? Whoops, kind of messy. So it's very similar to mitosis in a lot of ways, except for the tetrads that form. What are tetrads? They are homologous pairs. And we talked about homologous pairs yesterday. Homologous pairs code for the same types of genes, like eye color, but not the same one necessarily, right? Now imagine there's like 10,000 genes or thousands of genes on each of these chromatids. Now you start to understand how complicated this gets. The next one we said was metaphase 1. Metaphase 1, something else happens here, something very unique. My chromosomes line up in the middle. M's for middle, right? We remember that much. Now at this point, I know I have these all the same color, like all yellow and these all green. Would they be all the same? Would each of those be different? They each be different, but for just ease of drawing, I'm not doing that, okay? On the ends of each cell, I would have my centrosomes, which we all know is two centrioles, two centrioles in each centrosome, each with spindle fibers that will be attached to the centromere. Except you'll notice something. There's a left and a right side of that cell. Spindle fibers are coming in. Do I have spindle fibers from the right side connecting to the chromosome on the left? No. That's unique. So because of that, and because they're these two chromosomes are holding on to each other, they line up side by side, but not vertically. Remember in mitosis we did this? Oh my gosh, why does my computer keep doing that today? Remember in mitosis it was like this, down the middle? Remember that? That doesn't happen in meiosis 1, okay? In metaphase 1. In metaphase 1, they line up side by side. Why? I'm going to cut my genetic information in half because I'm making a gamete, a sperm and an egg. I'm not making a somatic cell. So they line up side by side. This is where the law of independent assortment comes in. So this is where we get the law of independent assortment coming in. Why? This means that did I say which of those chromosomes goes on the right? Like you go on the right, you go on the left, or did they just do it randomly? They just do it randomly. Okay? This is going to lead more into chapter 12 when we talk about Mendel's laws. But the fact that I don't say you go here and you go here, because remember they're all different, and you go here and you go there, means that they do this randomly. Does this create even more variation in who the future you will be? Yes. This makes not only each chromatid different, but the arrangement of how we put those in together different later, right? I mean, think about it. 23 pairs of chromosomes, 46 chromosomes splitting in half in complete randomness. All right, back to notes for a minute. So in anaphase 1, we know after metaphase it is always anaphase, A for apart, right? The homologous chromosomes separate and the spindle fibers pull those chromosomes of each pair to opposite poles of the cell, but the chromatids do not separate, right? We keep the chromosomes separate. Each chromosome is still made of 2 chromatids, and the genetic material has been recombined. So let's take a look at that. Let's go back to our drawing board. So anaphase 1, we still have our centrosomes, which are 2 centrioles. We know that. On both sides of the cell, we understand that there is a chromosome getting pulled each way, which has 2 chromatids. Oops. Was that one of my colors? It was? Okay. What was I doing making the green the same color? Okay. So what that looks like is something like this, okay, where we now separate. Now at this point, I basically have one N here and one N here, right? You get that? Okay. Anaphase 1. Now during telophase 1, the cytoplasm divides through cytokinesis, and the 2 new cells are formed, but both cells have 1 chromosome from each pair of homologous chromosomes. What does that look like? Well, so at this point, I now have separated my chromosomes, cutting my chromosome number in half. So I'm no longer a diploid germ cell. I am now a duplicated chromosome haploid cell. Okay. Now here's the thing, guys. Why can't I stop here? I still need to do what? Separate my chromatids, right? I need to get down with just one chromatid, because right now they're duplicated. That doesn't work. Okay? When we come to combining 2 gametes, we need to have just one of those chromatids. So we're not done yet, are we? So meiosis 2 begins with the two cells that form at the end of telophase of meiosis 1, and the chromosomes are not copied between meiosis 1 and meiosis 2. Why don't we just copy them again? We'd be right back to where we started, and we have to go through meiosis 1 again. We just never-ending cycle, right? So in prophase 2, spindles form. So you mean we're going to have another prophase? Yes. So in prophase 2, so in prophase 2, you're going to have two cells doing this, right? I'm not going to draw two cells doing it. You want me to draw two cells doing it? Just one? Is it okay if I just do one? All right. Well, let's do two. I don't want to confuse anybody. Maybe we'll switch to one in a minute. So in prophase 1, any nucleus that's there would have started to break down. My centrosomes never really went away. They'll still be there. Sometimes the nucleus doesn't even reform in between the division. Spindles start to form. And of course, I have my chromosomes inside of each cell, getting ready to divide, right? Okay? Do they pair up in homologs here? Say there are no homologs, right? Yeah, we separated, remember? We separated them during anaphase 1, sorry. So, a lot of stuff going on here. Very similar to any other prophase. Except we're doing this with 1N cells, haploid cells. We good here? So during metaphase 2, the chromosomes line up along the equators and are attached at their centromeres to spindle fibers. So nothing new. This looks just like mitosis in a way. So this is metaphase 2. And this is, of course, happening twice. So we understand that now things do not line up side by side. They don't have partners anyway. They're going to line up down the middle, right? And this is where things start to look an awful lot like mitosis with two cells and half the number of chromosomes. Metaphase 2. Happening in two cells. Both cells are 1N. What's the next step, do you think? Pull them apart, right? Anaphase 2. Okay. So during, we did that one. So in anaphase 2, the centromeres divide and the chromatids, which are now called chromosomes. Well, remember, after anaphase happens, we say they're their own chromosome. Okay, and they move to opposite poles. What does that look like? So in anaphase 2, must be a glitch in these boards. I don't know why that keeps doing that. Anyway. Does this get a little tedious? Yes. Oh, I forgot one. So in anaphase 2, I separate my chromatids. Now I have two 1N cells still, right? But they will eventually each have their own set because each of these is considered 1N because they now have a set of chromosomes, okay? Only one set. So when we finally do get into the last telophase, the nuclear envelope forms around each set of chromosomes, the spindles break down, and the cell goes through cytokinesis. And the result of meiosis is 4 haploid cells. So that means you got to do one more. Telophase 2. All right, here we go. I don't know what that is. So at the end, look like giant eyes. At the end, we have 4 1N cells which will eventually grow a flagella, right, if they're sperm. Okay, and that's what we're talking about doing here, right? What questions do you have about meiosis? It's very similar to mitosis in a lot of ways, but has differences. Very easy to get things mixed up, right? Very easy. Read test questions carefully. So, essentially this is what I just drew, but with only 2 pairs of chromosomes. So let's just talk about it generally. First stage, okay, we start off, we are definitely a 2N cell at the beginning, right? Our chromosomes become visible, nucleus starts to break apart, centrosomes show up with centrioles and spindle fibers, and they form tetrads in homologous pairs. This is where crossing over occurs. What is crossing over? Where 2 homologous pairs switch parts of their neighboring chromatid. This gives us more genetic diversity, increases biodiversity as a species. In metaphase 1, they line up side by side. Did I say who goes on the left and who goes on the right? No. That's where independent assortment happens at. Please remember those 2 things. Crossing over, prophase 1, meiosis 1, independent assortment. Okay? Anaphase 1, they come apart. When I say they come apart, I mean chromosomes, not chromatids, because they're lined up side by side. And in telophase 1, we have 2 new cells now, each with basically 4 completely different chromosomes in this case. Okay? Then we go through another division. We go through prophase 1. It's just like mitosis. Chromosomes are still visible. Nucleus breaks down. Centrosomes head to each side. Metaphase 1, they line up in the middle. Now single file, not side by side. Spindles start to tug on them. And in anaphase 1, we separate the chromatids, now each their own chromosome. So at the end of prophase 1, we had 1N cells. And then when we finally reform each of these new ones, they are still 1N. That's how we get 4 from 1. By only cutting the genetic information in half with the first division. Okay? I don't like sure. Sure sounds iffy. So I'm going to stop here for a minute. All right? And we're going to do a little project. Just a little one.